Welcome to this explainer. I am absolutely thrilled you're here today because we're going to dive into one of the most intense, high-stakes environments in the entire modern business and scientific world. We're talking about the gauntlet. That's the brutal, multi-level obstacle course every single new medication literally has to survive before it ever reaches your medicine cabinet. From an executive perspective, this is a multi-billion-dollar pipeline that is just fraught with operational risks where a single misstep can cost a pharmaceutical company an absolute fortune. So, it all begins with a pretty fundamental question. How does a chemical actually become a cure? It's not just some lucky accident in a lab. It's a highly structured, fiercely regulated journey designed specifically to weed out the weak and protect the public. Think of it as a series of strategic gates. At each one, core actionable decisions have to be made about whether to continue pouring capital into a project or to just pull the plug entirely. Okay, let's map out our roadmap. Our molecule has to conquer 5 specific milestones. Phase 0, the initial microdose. Phase 1, the first time it enters humans. Phase 2, the crucial efficacy test. Phase 3, the massive pivotal trial. And finally, Phase 4, the post-market watch. This chronological journey scales all the way up from that initial exploratory stage right through to post-market surveillance. And it's important to remember that every single step forward represents a massive, massive jump in both the data gathering requirements and the financial investment. All right, let's kick things off with Phase 0, the microdose. Strategically speaking, this phase is just a brilliant way to gather early intelligence with relatively low capital exposure. Now, you might assume these first-in-human trials are looking for safety or efficacy, right? Actually, no. That might sound a bit counterintuitive, but at this point, which is governed by the FDA's 2006 exploratory IND guidance, the goal is purely reconnaissance. Before committing massive resources to full preclinical toxicology, researchers really just want a sneak peek at how the drug behaves. To do this safely, they use sub-therapeutic microdoses. We are talking incredibly tiny amounts here, less than 1/100th of a calculated active dose. They use advanced techniques like PET imaging to literally watch the real-time distribution of the drug in the body, or accelerator mass spectrometry to detect carbon-14 at mind-bogglingly small levels. So the entire goal here really comes down to 2 heavy jargon terms that we need to demystify right away: PK and PD. Pharmacokinetics is simply how the body processes the drug. So does it absorb it? How fast does it excrete it? Pharmacodynamics, on the other hand, is how the drug affects the body. Gathering this early PK and PD data is absolutely crucial for helping executives and scientists make that very first go/no-go decision. Moving right along to Phase I, first in humans. The exploratory reconnaissance is over, and the stakes get very real. Notice the huge jump in scale here. We're now testing on 20 to 100 healthy volunteers. Well, a quick caveat to that, if it's an oncology drug, like a cancer treatment, they actually bypass healthy volunteers and go straight to patients with the disease, simply due to the nature of those specific medications. The primary operational focus now shifts entirely to establishing a safety baseline. This is a critical risk mitigation phase. Basically, you want to uncover any major safety flaws right now, rather than after you've invested tens of millions of dollars into further development. To establish that safety profile, researchers have to find the MTD, which stands for the maximum tolerated dose. Essentially, doctors have to find the ceiling. What is the absolute highest dose a human can handle before the toxicity becomes unacceptable? Now, to find that ceiling without putting people in unnecessary danger, they use what's called the 3+3 dose escalation design. It's kind of like testing the weight limit of a bridge by driving cars onto it one by one. You give 3 patients a low dose. If one shows dose-limiting toxicity, or DLT, you cautiously add 3 more patients at that exact same dose. If 2 or more out of the 6 hit that toxicity limit, you immediately stop escalating. It is a highly structured, very mathematical approach to operational safety. Next up, Phase 2, does it work? Assuming our drug doesn't get flagged for unacceptable toxicity, this is the crucial hurdle where we finally ask the million-dollar question: does it actually treat the targeted disease? And the scale escalates significantly again. We are now looking at 100 to 500 patients. And crucially, unlike Phase 1, these are patients who actually have the targeted disease. This is the very first real efficacy test. Why? Well, because animal models, which are used extensively before human trials even begin, often completely fail to predict how a human disease will respond to a drug. You can cure cancer in a mouse 100 times over, but the human body is just vastly more complex. So here is the brutal truth. Phase 2 is an absolute bloodbath for new drugs. Compared to industry benchmarks, this phase boasts a massive, pipeline-crushing 67% failure rate. 2 out of every 3 drugs that enter Phase 2 are going to fail right here, usually because they simply don't work as hoped, or the required dose to make it work proves way too toxic. For executives, this is the ultimate operational risk hurdle. Because the failure rate is so incredibly high, companies employ strategic trial designs to filter out the duds as efficiently as possible. You've got your randomized controlled trials, which are the gold standard for sure, but you also have really clever adaptive designs like Simon's two-stage. Simon's two-stage is fantastic from a business perspective because it includes a built-in futility analysis. Think of it as an executive kill switch. If the drug fails in stage 1, the trial stops early, saving millions in wasted capital. All right, the final boss before approval. Phase 3, the pivotal trial. If a drug actually beats those Phase 2 odds, it enters the most demanding hurdle yet. The numbers here are just booming. This level requires massive scale, 1,000 to 3,000 plus patients spread out across multiple clinical centers, sometimes even globally. As you can imagine, the capital expenditure here is astronomical. The goal here is therapeutic confirmatory. Regulators like the FDA rely entirely on these definitive, adequate, and well-controlled trials to finally confirm both the efficacy and the safety profile required for official market approval. Generally speaking, the FDA wants to see data from at least 1, and very often 2, of these massive pivotal trials just to ensure the results aren't some sort of statistical fluke. Now, to ensure these multi-million-dollar trials yield rock-solid, unbiased results, they employ strict blinding levels. In open label, everyone knows who gets the real drug versus the placebo. In single blind, the investigators know, but the patients don't. But the absolute gold standard is double blind. Neither the patient nor the doctor administering the dose has any idea what it is. This rigorously strips away the placebo effect and investigator bias, protecting the integrity of the data that executives will eventually take straight to the FDA. But approval isn't the end. We enter Phase 4, post-market watch. You might think FDA approval is the finish line, right? But the process actually continues as the drug enters the real world. Post-marketing surveillance is incredibly rigorous. The FDA heavily monitors drugs post-approval by issuing legally mandated post-marketing requirements, or PMRs. This is a massive compliance risk for a pharmaceutical company because failure to meet these ongoing monitoring requirements can result in the drug being forcefully withdrawn from the market entirely, wiping out all that prior investment, which is exactly what happened historically with the painkiller Vioxx. They track safety through pharmacovigilance systems like FAERS, where adverse events are spontaneously reported. And for drugs that have significant safety risks but are still highly effective, the FDA uses something called REMS, Risk Evaluation and Mitigation Strategy. This basically means the drug is kept on a very tight leash. The company might be forced to restrict distribution only to specially certified pharmacies, or mandate that patients enroll in specific, tightly controlled registries. What's really fascinating is how we gather this data today. Phase 4 relies heavily on real-world evidence. This doesn't just come from doctors' offices anymore. It's pulled from electronic health records, Medicare claims databases, and, increasingly, wearables and digital health data. Which means if you're taking a newly approved medication and wearing a fitness tracker, you yourself might unknowingly be participating in Phase 4 data collection. Which leaves us with a truly incredible thought to end on today. As the boundaries between technology and biology continue to blur, how is your own smartwatch shaping the future of medicine right now? The gauntlet doesn't end in the lab. It continues every single day all around us. Thank you so much for joining me, and I really hope this explainer provided a clear, actionable look at the incredible journey of drug development.